Alright, so before we can get into the process of DNA translation, we first need to understand how tRNA is charged. So basically what happens is tRNA synthetase, which is an enzyme, um, read, understands what the anticodon is for that particular tRNA and chooses the specific amino acid that needs to bind to that tRNA anticodon. So basically how this happens is the enzyme gets the amino acid and it allows it to bond, which now forms an AMP complex. All right. So the original energy used is ATP, and when it becomes an AMP, two phosphate groups are lost. So that means that energy's gone and it is now AMP. So now later on in the reaction to form the polypeptide chain, this this energy is used to create the bonds and keep it strong. All right. So for DNA translation, the first thing that happens is the small subunit of the ribosome comes along here, the mRNA, until it reaches AUG, which is the start codon, which codes for the anticodon of UAC and the amino acid of methionine. All right, so now that we know where methionine is, the large subunit of the ribosome comes in here and comes together basically, and the methionine and the tRNA enter into the P site. Now, once we got here, the ribosome keeps moving along until it gets to the next tRNA section and the amino acid, which then enters in through the A site, which now the amino acids covalently bond, and it translocates to the P site, and the first tRNA goes to the exit site and then exits completely. And now this continues along this whole chain here until eventually it gets to the stop codon, which codes for a release factor, which the release factor comes in here and breaks all of this up, leaving just the polypeptide chain right there. And that is DNA translation.